Welcome to this video in which we will see about the study of lactose operon practically. So lactose operon is expressed when lactose is present in the medium. So lactose operon expresses the gene for taking lactose inside the cell, that is by enzyme lactose permease, and the second enzyme which operates is beta galactosidase, which breaks down the lactose into glucose and galactose, which can be further utilized to obtain energy and for biosynthetic purpose. So basically lactose operon is for utilization of lactose sugar present in the environment of the bacterial cell. So if you want to check the expression of lactose operon, you can have three different flasks. The first flask will be having basal salt medium with glucose in it as a single carbon source. A second flask will be having basal salt medium with lactose as a single carbon source, and the third flask with basal salt medium and glucose and lactose as two different carbon sources in the medium. In the first flask, where you will be having glucose as a single carbon source, since the lactose is not present, culture will not go for expression of beta galactosidase gene or expression of lac operon genes, and that's why operon will be shut off, and the glucose which is present, it will be utilized by the culture for the growth and development purpose. In the second flask, where you are having lactose, since the lactose is present and no other simple sugar is present, the culture needs to utilize that lactose for the growth purpose, and for utilization of lactose, culture will produce lac permease enzyme, which will take the lactose inside the cell, and beta galactosidase enzyme for breaking the lactose into glucose and galactose. So the operon will be on in this second case, and the lactose will be utilized and culture will grow in that particular flask. In the third flask, where you are having glucose and lactose both carbon sources, since the glucose is monosaccharide, it is easier to degrade and obtain energy from it. The culture will prefer to utilize glucose first, and in the initial stages the culture will not express the lactose operon. So in initial stages, glucose will be utilized for the growth purpose. Once the glucose is utilized, a culture will take a lag phase, in which culture will produce enzymes which are required for taking lactose inside the cell and for utilization of lactose. Once these enzymes are produced and the genes are expressed, culture will start utilizing the lactose as a carbon source, and again it will start multiplying. So you will be having second exponential phase. Once the lactose is utilized, you will get stationary phase, where the death rate and the formation of new cells, rate of formation of new cells will be equal because of accumulation of toxic products and utilization of nutrients. The number of cells will remain constant, and then once the components are utilized and toxic components are accumulated, the cells, they will start entering in the death phase. So you will get a diaxic pattern in the flask, which will be having glucose and lactose both carbon sources. So in the third flask, you will observe that glucose will be utilized first, and then lactose operon will be operative, and then lactose sugar will be utilized. Thank you.